Hello! I am going to try to finish this chapter with this video. So let's go quick. Regulation of hexokinase. So all of this slide is basically a review of what we've already talked about. Hexokinase is inhibited by glucose 6-phosphate in muscle cells, in liver cells. There's also glucokinase, which is an isoenzyme. Higher Km for glucokinase means it needs a higher amount of glucose before it will catalyze the reaction. So finish reading that slide and then we'll go on to the next. So how about regulation at the end of glycolysis, pyruvate kinase? All of these we already listed in our table, so I'll go on. So here's something new. Pyruvate kinase comes in both an M form and an L form. Those are two different isoenzymes. So again, just like hexokinase, where we had two isoenzymes, we have two for pyruvate kinase as well. So the M isoform is found in the muscle. The L isoenzyme is found in the liver, L for liver. So the liver pyruvate kinase can be phosphorylated, and the phosphoryl group slows down the enzyme, inhibits the enzyme. I kind of think about that as, like, let's pretend that the phosphoryl group is attached right by the active site. So the phosphoryl group inhibits the substrate from getting into its binding site, slowing down the activity of the enzyme. So how does that get phosphorylated? The enzyme gets phosphorylated by a protein kinase called protein kinase A. And protein kinase A gets activated by a cyclic AMP cascade, which is triggered by the hormone glucagon. So glucagon is kind of opposite of insulin. Insulin is a hormone that signals to the body that there's a lot of glucose in the blood. Glucagon, on the other hand, is produced when the glucose sugar level is low. So if there's low glucose in the blood, in other words, a person's fasting, they haven't been eating carbohydrates, the blood sugar level goes down. Glucagon is produced in the body. It triggers the cyclic AMP cascade that turns on protein kinase A, which can phosphorylate pyruvate kinase and inhibit it, make it less active. So this diagram shows phospholinopyruvate plus ADP going to pyruvate plus ATP, and the dephosphorylated form of pyruvate kinase is active. When it gets phosphorylated, it becomes inactive. So let's go ahead and talk about one of the last sections of this chapter, transporters for glucose to get in and out of the cell. So these transporters are called GLUT1, GLUT2, GLUT, and they're a family of transporters. All of them span the plasma membrane with 12 transmembrane alpha helices. So they have like sort of a pore where glucose can go through. And they're sort of like channels, but they're not channels, because glucose can go either way, either out of the cell or into the cell, depending on the concentration gradient. If there's a lot of glucose outside of the cell, the glucose will be transported into the cell. If there's a higher concentration of glucose inside the cell, then the transporters will transport glucose out of the cell. Each cell has a different type of transporter, 1 or 2 or GLUT3 or GLUT4 or GLUT5. Actually, there's over a dozen of these, but our book luckily only tells us about 5 of them, because different cells have different jobs to do. For example, liver cells, you know, have the job of storing glucose and doing synthesis of other molecules. Pancreatic beta cells, you might remember, make insulin and secrete insulin to indicate to the body that the glucose level in the blood is high. Well, both liver cells and pancreatic beta cells have GLUT2 transporters. And GLUT2 have high K sub M's. Those are the Michaelis-Menten constants. And if it's got a high K sub M, that means that they will only work, or they'll work at a reasonable velocity when there's a high blood sugar level. So high Km means they need a lot of their substrate before they will go at a very good velocity. Okay, so let's go on. Oh, here's a table of the family of glucose transporters. All mammalian tissues have GLUT1. They may not have very much of it, and that's the basal glucose uptake. GLUT2 is liver and pancreatic beta cells, and look, they have a very much higher K sub M. And the GLUT4 is for muscle and fat cells, and that's kind of a medium Km. Let's go on to the last section of this book, section 16.5. And this talks about insulin again. Insulin is a polypeptide hormone. It's secreted by the beta cells of the pancreas in response to high blood levels of glucose. And the function of insulin is to stimulate the uptake of glucose by other tissues, such as muscle and adipose tissue. So going on to the last slide here, this is a pancreatic beta cell, and this is to explain how pancreatic beta cells know that the blood glucose level is high. Notice that this pancreatic beta cell has a nucleus, and it has a vesicle towards the right side with insulin in it. It makes insulin, and something triggers the vesicle to fuse with the plasma membrane and to release the insulin to the bloodstream, to go to all the other cells of the body to tell them it's high blood glucose level, take glucose into your cells. So let's start with the upper left corner here. The pink transporter there transports glucose into the cell. Glucose undergoes glycolysis and cellular respiration. So that's citric acid cycle electron transport chain oxidative phosphorylation to make ATP. So ATP level goes up as glucose comes into the cell. Now, there's the green one is a potassium channel that allows potassium to flow out of the cell. As the ATP concentration goes up, it actually can inhibit this potassium channel, so stopping potassium from leaking out of the cell. So there's more potassiums inside, more positive charges inside, upsets the ion concentration and changes the ionic charge across the cell membrane. The blue channel is a calcium channel that allows calcium to flow into the cell only when it's triggered by the change in the ionic gradient across the cell membrane. So as ATP blocks the potassium, that changes the the voltage or the ionic strength across the membrane and opens the calcium channel. Calcium flows in. It interacts with the vesicles containing the insulin to cause them to fuse with the plasma membrane, releasing the insulin. So increase in ATP closes potassium channel, alters the charge across the cell membrane. This alteration opens the potassium channels. The influx of potassium ions stimulates the release of insulin into the blood. And that is the end of this chapter.